In this video we're going to look at a dataset that has previously been calibrated in terms of energy using the chlorine 2p doublet, and the chlorine 2p 3/2 peak has been aligned based on a peak model so that all of these data now have been shifted based on the chlorine 2p on a row by row basis. The motivation for calibrating these data is so that all of these spectra are then aligned in terms of binding energy, taking out the influence of charge compensation that was performed during the acquisition. And the reason that we want to do this is so that we can make use of options on the calculator property page, the PCA property page, and the test data property page that'll help to understand the chemical changes that are occurring as a consequence of this particular experiment where the surface is being modified by UV radiation. One of the consequences of charge correcting spectra is that data bins now are shifted relative to one another, so they don't line up anymore. And if we're going to do any processing that involves principal component analysis, or the other one is merging data in a regular sense, then what we'll need to have is all of these bins reassigned and realigned so that we have a single start and increment for each one of these spectra, even though at this point that's not true. And we can do this using the calculator property page, and there's an option here that says rebin selected VAMAS blocks. So if I select these VAMAS blocks and then I press the rebin, then what happens is if I display these chlorine again, you can now see that the data set has been modified so that each spectrum starts at exactly the same energy and the same increment is used for each one of these spectra. So the idea of performing this operation is that if we want to do any type of vector processing, such as the principal component analysis, or if we want to do any kind of different spectra, we now have data in a form where each data bin aligns and is open to that type of analysis. A new file has been created that contains these rebin spectra, and we will start by displaying each row one per tile and go to the test data property page, and we'll create merged regular spectra. The idea of regular means regular intervals in energy have been used to acquire the data, and the data will then be collected out of each one of these display tiles. And you can see in this scroll list we've got each one of the rows in the right hand side now appear overlaid in these display tiles, and you can see the relationship that the relative intensity of these peaks is based on the photo emission process and the response of the instrument, and also these peaks are spread out in kinetic energy, so they are over quite a wide range, and it's not easy to see at a glance all of these individual smaller peaks. And so we're going to use the merge regular, which will take the information in each tile and create a set of VAMAS blocks that are all written back to the same file and now appear in a slightly different form, in that the data has all been concatenated in terms of energy. So the energy scale is no longer applicable, at least for most of these data. One of them will be correct, that'll be the first one, but all the others, this is an arbitrary scale, so we have to indicate which peak is which by labeling these using annotation. So these are all labels that have been added automatically to the data by the process of merging these spectra in a regular sense. And the reason that we would like to do it this way is it means that we can see at a glance how different peaks have evolved as part of this measurement. So here we have two different measurements, and for example you can immediately see here that there's been a change in the nitrogen peak, and also if you look at the carbon, there's been a change in the carbon peak as a consequence of irradiating the surface with UV light. Simply inspecting these carbon 1s spectra, you can see that things have changed. If you consider the relationship between how peaks have evolved in the one carbon 1s with respect to the nitrogen, for example. You might also consider how an oxygen peak is evolving with respect to the carbon 1s and the nitrogen. While it's useful to compare in this way, these merged data, the other advantage of merging these data and having them in this particular form is that we can construct different spectra. And choosing two spectra that have a wide variation, such as these two here, means that I can now look at how different proportions of these two spectra, when subtracted one from the other, produce a set of so-called different spectra that give an insight into how these different relationships evolve. So if I go to the calculator property page and I use the different spectra, and this relies on the bins aligning, we're simply going to subtract two sets of spectra where we're assuming all the data bins are aligned and have an equal step size. So this is why we had to rebin the spectra before performing this calculation. When I say different spectra, a new data file is created, and within this data file we have a list of spectra that have all been calculated from the two spectra that were overlaid in the original active tile. And the idea is that the first one and the last one in this list are those two spectra. And then as we move somewhere within this sequence, we obtain different spectra that don't necessarily look physically meaningful, but they give us some insight into what's going on. For example, here we have a negative going peak associated with the nitrogen 1s, and this seems to be correlated with a particular peak in the carbon 1s, which has also gone negative. So let's start moving around this in a systematic way and see how these peaks evolve. So this is not a real spectrum. This is a spectrum that has been calculated from two spectra, and as a consequence one of the nitrogen peaks is no longer within these data, and the shape of the carbon envelope has changed, and so too has the oxygen 1s. What is more interesting from the analysis of this data set is that a feature has emerged within the valence band that wasn't as obvious in the original data set, and we can correlate all these things, such as the feature here, with the shape that we now see in the titanium 2p. So we see this shape here represents the emergence of a 3+ state of titanium, and this peak here looks more like a 4+. But what is more intriguing in terms of what's possible by this technique is that we've ended up with a sequence of carbon peaks that we might use to construct a peak model. We have a nitrogen 1s peak and a much simplified oxygen 1s. So I'm going to take a copy of this. So I now have a copy of the different spectrum that I found interesting, and I will now start searching to see what else I can find. So having searched through the list, I come up with another example of a spectrum that I find interesting. That is that I've got a completely different shape now for the carbon 1s. The oxygen 1s has additional structure here that it didn't have before, and I've now got two peaks in the nitrogen 1s, plus the titanium now looks like a 4+ state, and the valence band is similar, but it has a small structure here, but it's not the structure that we saw when the spectrum looked more like there were 3+ in the titanium. So we'll save this one too. So the outcome of merging VAMAS blocks and then doing a difference analysis are a pair of spectra that give you a different perspective of the data set other than the one that was originally presented by the measured data. So from these different spectra, there's an indication that you have at least one form that might be a 4+ state for titanium, and there's another form that looks like some type of combination of a 3+ and some 4+ like feature within the spectra. And this would be difficult to see from the original data, and by doing the difference data we can separate this type of information. So a further example would be to say, look at the oxygen. So we've got a peak here that looks like it's the oxygen 1s, and it seems to be correlated nicely with a large carbon peak bonded to oxygen, and you can clearly see that the shape of the carbon 1s spectrum is very different from anything that we saw in the original data set. These two component vectors represent separation of chemical state information using the difference approach. Once we have a pair of components, we can go back to the original data and we can then perform a least squares analysis of the original data, making use of these two spectra. So we're going to add these to the selection, and then we'll copy them through to a new file. And what this does is it puts the data that we want to analyze in terms of these two components into the same file, so we can overlay in the active tile the two component vectors where we've got this separated chemical information, and go back to the original data. So I'm simply selecting these, bringing up the spectrum processing dialog window, principal component analysis property page, and then I'm going to press the generate spectra button. So this button acts on the information that's overlaid in the active tile. These represent the basis functions, and these selected VAMAS blocks represent the target functions. So I can now press this button, a new experiment frame is created, and the experiment frame contains two components here. These are the component vectors, and these component vectors have been scaled in a least squares sense, so when they're summed we end up with this vector here, which represents the least squares solution. And when I then add in the first column, I've got a least squares solution summed together to give an approximation to the data, and the data appear in the first column. So let me just reorder these so that the colors are as normal, which would be black first, green second, and then we have blue and then red. You can see this from the color property page. So it goes black, green, blue, red. That's the order of selection of these VAMAS blocks. So we've got black is the raw data, green is the least squares approximation, blue and red are the components, and these have been calculated for each one of these spectra in the first column. So as I step down these rows, we're getting a different decomposition based on the spectral information, and you can see that the sample is evolving and changing between these two states in a very systematic way. So this represents one way of observing the trend within these data, and you could then plot various information that you calculate from these individual components to give you more information about these samples in terms of these two component vectors.